In this video, we'll look at the Antibody Component Analysis data function in ChemCharts. The Antibody Component Analysis data function provides a way to compare regions of antibody sequences and relate those differences to define properties of the antibody. For this example, I have a table of 242 commercial antibody sequences from a paper by Revvity et al., and I have run the antibody numbering data function to generate annotations of the framework and CDRs for each antibody. I have also created an activity column of random numbers to use as data for this example. To create the sequence column inputs for the data function, each region of interest is extracted to its own column using the extract GenBank features data function. This data function creates new columns for each annotated feature or region in the GenBank representation. These individual columns can then be used as inputs for the Antibody Component Analysis data function. The older extract GenBank regions data function will also do this, but extract GenBank features is recommended. For more information about the extract GenBank features data function, see the Working with GenBank Files video and refer to the Data Function User's Guide. From the Data Function Settings dialog, choose the data table containing the sequences of interest. Next, select the sequence columns for which comparisons are to be made. Here, I will select the columns containing heavy chain CDRs that we extracted previously. With these selections, the data function will look for pairs of sequences that are identical for 2 of the 3 heavy chain CDRs. The differing CDRs, their sequences, and the difference in activity between these rows will be gathered into a table. It is best to keep the number of selected regions fairly low, as it may be difficult to find identical sequences across a large number of regions. Select a column containing a unique ID for each row. It is important that these IDs are unique in order to annotate differences in activity in the results table. The data columns can be one or more numerical columns containing data related to the sequences. In this case, I'll use the activity column of random numbers that I generated previously. The gap mismatch penalty is used when aligning different sequences from the same region and will contribute to the overall alignment score. -1 typically performs well in most cases, but increasingly negative numbers will discourage gaps in alignments. Once the data function is finished, click the Configure Visuals icon to create the results table visualization. Each row in the results table contains information about the comparisons made between sequences from the original table. For example, for the first results row, source column contains aligned sequence CDR-H1. This means that the results in this row are from a comparison of CDR-H1 regions for sequences that had identical CDR-H2 and CDR-H3 regions. LHS-ID and RHS-ID are the IDs for the sequences that were considered the left-hand side and the right-hand side sequences during comparison. In this case, the left-hand side is satralizumab and the right-hand side is tocilizumab. The LHS sequence and RHS sequence columns contain the CDR-H1 sequences for satralizumab and tocilizumab. The sequence pair column shows the alignment of the two sequences. The top sequence is the LHS sequence and colored using ClusTAL2. The lower sequence is the RHS sequence and colored with the Blossom comparison color scheme. The Blossom score is the alignment score for this alignment. The activity column, corresponding to the name of the original activity column, shows the change in activity from the LHS value to the RHS value. Putting all of this together, row 1 in the results table shows that when comparing satralizumab and tocilizumab, their CDR-H1 regions differ, while their CDR-H2 and CDR-H3 regions are identical. The change in the CDR-H1 regions are shown in the sequence pair column, and they have a difference in their activity values of 7 units. This number is positive because the LHS, satralizumab, has a higher activity than the RHS, tocilizumab. Remember, the activity values here are just random numbers used as examples. The remainder of the table has 14 rows for comparisons in which CDR-H3 is different, while CDR-H1 and CDR-H2 are identical. Thanks for watching, and check out our other ChemCharts tutorials.